In this video, we are going to learn about the extensions that go beyond Mendelian genetics. Okay, so the first thing we're going to talk about is degrees of dominance. So in Mendel's experiments, all of the characters have what we call complete dominance. This is where the phenotype of a heterozygote and the dominant homozygote are the same. So with both dominant and heterozygous pea plants, if they have a purple flower, right, versus a homozygous recessive plant would have a white flower. So let's just write this out. So capital P would be purple. But heterozygote, capital P, lowercase p, would also be purple. Whereas lowercase p would be white. This is complete dominance, right? But the second thing, degree of dominance, that we're going to talk about is what we call incomplete dominance. So with incomplete dominance, neither allele is completely dominant over the other, and the heterozygous offspring would have a phenotype that's kind of somewhere in between the parents. So let's take a look at the example on the right. For this flower at the P generation, so we're still up here, right? Two true breeding plants parent. Homozygous dominant is red and homozygous recessive is white, right? And this is a result from a functional allele that codes for that enzyme to produce either the red pigment or the other allele that does not produce a pigment, which is why they end up white. However, in the F1 generation, where all the offspring are going to be heterozygous, because remember we have, let's call this capital R, and lowercase r. And then when we come down here, we would have all heterozygotes. And instead of being red or white, they appear pink. And that's because they only get one allele that makes the red pigment producing enzyme. So as a result, they're going to produce less red pigment compared to the red parent up here. Therefore, they are going to appear pink instead of white. So this might kind of sound like the blending hypothesis, which states that the traits of both parents blend together in the offspring, but it actually doesn't support this, because when you go into the F2 generation, you'll see red and white both appear again, because now we have the chance to have homozygous dominant or homozygous recessive again once we have our heterozygotes breeding. And that would lend to, this isn't actually a blending hypothesis, it just, this trait shows what we call incomplete dominance. Okay, the last type of dominance is called codominance. So codominance is when the two alleles affect the phenotype to the same power. Okay, so if we look at the example on the right, let's say we cross a red fish and a blue fish, and then out comes a spotted red and blue fish. And this is probably because for the character of body color in this fish, one allele is coding for enzymes to make the red pigment and another is coding for enzymes to make the blue pigment. So if we have a heterozygous individual, both types of enzymes are working together. Therefore, you can see both colors on the body. So this is different than incomplete dominance. Incomplete dominance is kind of that you have that in-between stage, right? Example, red and white combining to make pink. And here we have codominance where both of them are still showing up. The other thing you need to know is that most genes actually exist in more than two allelic forms. Although each diploid individual can carry up to two different types of alleles, since we only have two copies of each gene. So here's like a perfect example of a gene that comes in multiple alleles, which is blood types in humans. So human blood groups exist in three forms. We have dominant IA, dominant IB, and I, which is recessive. Okay, so normally we just call this A or B, and then I would be the recessive. But IA codes for type A carbohydrates, IB would code for type B carbohydrates, and recessive I codes for nothing. So IA and IB are completely dominant over the recessive I, and they are codominant to each other, right? So we see what they code for up here, and then down here in this bottom table, we see that the different genotypes. So if we have IA or IA recessive I, the phenotype, the blood group, is going to be A. However, if we have IA and IB, those are codominant, so they're going to show up together. We're going to have both the A and B carbohydrate. Similarly, if we have IB or IBI, then we would get a B type blood group. Okay, and if we are homozygous recessive, so we have II, that is when we get the O type blood group. Okay, so very different, and this is a case where we have multiple alleles, where we could potentially have different kinds of blood groups even though it's coding on the same gene. Up until now, we've only really been looking at phenotypes that are controlled by one single gene. However, there are also cases when the products of the gene will actually interact with each other, and the phenotype of one gene could potentially alter the phenotype of another gene. So this is what we call epistasis. One example that you see over here is the coat color of Labrador retrievers, and it's actually controlled by two genes. One controls the pigment production, and one controls the enzyme that sends the pigments into the hair. Okay, so the first gene has two alleles. So we'll say gene 1, this is our B, right? This is our pigment production. So dominant B would produce black pigments, okay? And then lowercase b or recessive b would produce brown pigments. In this case, we're showing complete dominance. So black is completely dominant over brown. So both homozygous dominant and heterozygous would be black. So if I put capital B, or capital B, lowercase b, both of these would be black. So only if I had lowercase b, would that be the brown coat. Okay. The other gene also has two alleles. This is for E, right? And this allows the pigments to be sent into the hair. Recessive E, our lowercase e here, that does not allow the pigments to be sent into the hair. So there's two, or I guess I should backtrack. These are also complete dominance. So the problem with these two genes is that if the dog is a homozygous recessive for pigment deposit, it doesn't matter if it's black or brown pigment being produced because no pigment can be sent into the hair at the first place. So as long as the dog is a homozygous EE, so if it's EE, it's going to appear white or yellow. Sometimes we say yellow labs, right? So this is what epistasis is. When the phenotype of one gene, in this case our gene E, right? I'm going to do an uppercase E there, can alter the phenotype of the other gene, which would be gene B in this case. So you can see it doesn't matter what the B's are here. If it has lowercase e, homozygous recessive E, it's going to be a yellow lab no matter what. It's not going to change. Versus these other ones, even if it has a recessive E, it's not going to matter as long as it does have at least one dominant, and that's going to send the pigments, whether it's black or brown, into the dog. So this is how you get this cross. If we had, say, two black labs here mating, this is the potential. You have 9 black to 3 brown to 4 yellow. Okay, and then finally, a single trait can also be the result from multiple genes that act on it independently. So the example we have here is polygenic inheritance. Okay. So in polygenic inheritance, a single phenotype is determined by the additive effects of many genes. Okay, so in this case, the character is said to be a quantitative character. In polygenic inheritance, although the character is affected by multiple genes, these genes follow the law of independent assortment. Okay, so they will sort independently of each other. A good example that you see here on the right is human skin color. So human skin colors are controlled by 3 different genes. So we have A, B, and C that are all controlling pigment production. Each of these genes comes in 2 allelic forms, dominant, which would produce the pigment, and recessive, which doesn't produce the pigment. So for a heterozygous individual with a genotype of heterozygous A, heterozygous B, right, and heterozygous C, they would receive 3 pigment-producing alleles, which are symbolized by these black dots. Okay. And then they would have 3 alleles that do not produce pigment, which are the white circles. So you'll see, based on this, how many alleles they have producing pigments and how many alleles they don't. And you see the more alleles they have producing pigments, the darker the skin color. The less alleles producing pigments, the lighter the skin color. And since the 3 genes follow independent assortment, each individual will be able to generate 8 different types of gametes. So from here, we could have A, B, C, right? All dominant. We could have all recessive, A, B, C. We could have A, capital B, lowercase c, A, B, capital C. So there's 9 different gametes that can come from these guys. Basically 9 different combinations of what they could potentially pass on to their offspring. So since they all either produce or do not produce pigments, we're just doing the black and white dots down here, right? So if both parents are of medium skin tone, so you see here that we have about half and half, like we were just showing, their kids can actually be either very fair skinned, so like up here, to medium to potentially pretty dark skinned, as we can see in this big Punnett square, right? Of course, the probability of each skin tone is very different, with the very fair or very dark skin being pretty rare at 1/64. And then the medium skin tones have the highest chances, right? So we can see what the odds are of these different phenotypes down here at the bottom of the Punnett square. Okay, so let's now talk about Mendel's law of inheritance within human genetics, using what we call a family pedigree or just a pedigree. So Mendel came up with this inheritance principle by breeding the garden peas. And then years later, there was another famous geneticist, Thomas Morgan, who would mate Drosophila, which are fruit flies. And he discovered that crossing over of chromosomes as well as the sex link, I'll back up. He discovered that chromosomes can cross over as well as you have sex-linked genes. And we'll learn more about him later, but the point here is that great geneticists really study genetics by mating specific individuals and observing the genotypes and phenotypes of their offspring to be able to uncover the pattern of inheritance. However, scientists cannot manipulate the mating of people, at least not anymore. So when we study human genetics, instead of actively pairing 2 individuals and waiting to study their offspring, we usually do it the other way around by backtracking a particular trait over generations using a family tree, right? And when we look at this family tree, we can use it to describe particular traits of parents and children across generations, and that's what we call a family pedigree. So here's our first example. You'll see that this family pedigree is tracking widow's peak, which is that V-shaped growth of hair towards the center of the forehead. Some people have it, some people don't. So let's take a look at the top of the figure and see the key. So usually in a pedigree, male family members are symbolized by squares, while females are symbolized by circles. Most pedigrees will also let you know which individuals are showing the trait. So in this case, the ones with purple squares or purple circles are the ones that are showing the trait. So in this case, the ones with purple are the ones showing the widow's peak. Okay. So from the first generation, which in this family pedigree we're considering the grandparents, both the grandpa from dad's side and the grandma from mom's side have widow peaks. So both mom and dad have widow's peaks. Okay. And only one daughter has it while the other doesn't. Right? So we can see what their genotypes here. The phenotype is that they either do or do not have widow's peak. But this particular pedigree will show you the genotype of each individual. And even without this information, we could still conclude that widow's peak must be a dominant trait because the two parents with widow's peak gave birth to children that both had it and didn't. If this was recessive, then that pair would only be able to give birth to recessive phenotypes. Because if it were recessive, then dad would have to have a lowercase ww, mom as well. So the only thing they would be able to give would be these lowercase w's, which means that all of their kids would have lowercase w's. But what's actually happening here is they are heterozygous, so they have one dominant, one recessive. And now their kids, if we wanted to do a little Punnett square, right? We would have WW, and then we would have capital W, lowercase w, capital W, lowercase w, and lowercase. So we would have one homozygous dominant, one homozygous recessive, and then two heterozygotes. So overall, those are the genotypes. Our phenotypes in this Punnett square example, we would have 3/4 would have widow's peak, while 1/4 wouldn't. Right? So when we look at these individuals, right? Grandpa from dad's side, grandma from mom's side, dad and mom, they all have to be heterozygous because not all their offspring have the dominant trait. So we do have those probabilities factoring in, right? Where we might have some that are recessive and we might have some that are either homozygous dominant or heterozygous. And when we get down here to the daughter, we don't necessarily know her genetic or her genotype because she could have pulled a capital W from mom and a capital W from dad. Sorry, the opposite, dad and then mom. And she could be homozygous dominant, or she could have pulled one from each that were different and she could be heterozygous. But we know that these are the two possibilities because she does have widow's peak. Versus over here, the daughter that doesn't have widow's peak would have to be homozygous recessive. So here's another trait in the same family. So for this one, we're tracking the ability to not taste PTC, which some of you may or may not have done in a lab before, but PTC is a bitter tasting compound that can be found in a lot of bitter tasting food like broccoli. So some people like to call it the broccoli hating gene. And again, as you can see here from both grandparents, right? We have our first generation, our grandparents here. Both grandparents from dad's side, they can taste PTC. And then we know that they would have to be heterozygous because if we keep looking down to dad's side, we can see that they have children that have two different phenotypes. Some of them have it, some of them don't. Versus grandpa on mom's side cannot taste PTC and grandma can. So grandma must be heterozygous too because again, they have children that also have two different phenotypes. So we see here dad and mom both can taste PTC. And again, one daughter can and one daughter can't. Okay, so we know that the parents must also be heterozygous. And again, for the one that can taste it, we're not quite sure what her genotype is because she could be either homozygous dominant or heterozygous. But we can go through this, so even if you don't know for sure, you can probably narrow it down to two choices in most cases, right? So if you don't like broccoli or cauliflower or bitter tasting foods, it's likely that you would also be homozygous dominant or heterozygous for this trait. If you do like them and they don't taste bitter to you, it's likely that maybe you cannot taste PTC And so you're probably homozygous recessive for this trait. Not a definitive classifier, but definitely the most likely scenario. Okay, so the two examples we just looked at were kind of fun genetics that are interesting to track. But not liking the taste of broccoli or having a widow's peak doesn't really matter that much, right, in the grand scheme of things. But pedigrees really go beyond these examples. They're, in fact, being used pretty regularly for genetic analysis to be able to track more severe genetic disorders and to predict the chances for a couple giving birth to affected children. So it could be affected children of, disorders that may be run in one or both of the parents' families. So genetic disorders that can run in the human population can be caused by either dominant or recessive alleles. So let's look at some examples of human genetic disorders that are inherited either recessively or dominantly. For recessively inherited disorders, they are usually caused by an allele that codes for either a malfunctioning protein or no protein at all. This type of disorder only shows up in homozygous recessive individuals, and many of them are born to parents who are what we call carriers of the disorder. So a carrier is someone who is heterozygous and has one dominant allele that can carry the good copy and one recessive allele that carries the defective copy. So usually the good allele is sufficient to make the carrier parent normal or at least functional enough to reproduce, right? So in some cases, you might not even know you're a carrier because you are quote unquote normal and you don't have this disease. And so it's hard to know that you're a potential carrier and could pass it on to your offspring. Keep in mind that depending on if this is sex linked or not, males cannot be a carrier for some things if it's sex linked, or I should say X linked, which we'll talk about later, but not anything to get too worried about now. Just know that some people can be quote unquote normal, not have the disease, but be carrying the genes for it. So, since some homozygous recessive individuals might be able to reproduce, although they're certainly going to be a lot more aware of the presence of the disorder, if the disorder is lethal, no homozygous recessive individual will be able to live to sexual maturity and reproduce. So, in the human population, because of that, most recessive alleles are pretty rare, especially if it causes lethal conditions that the individual won't be able to survive until reproductive age. That's, evolution at its finest, and nature's way to kind of ensure the survival of a species by minimizing things that don't work. However, the chances of passing on the recessive traits to offspring do increase greatly if the two parents are close relatives because they both have a higher chance to be carriers since the affected allele does run in the family. And that's why in many countries the law prevents close relatives from getting married. Here's one example for a recessively inherited trait in humans, which is albinism, or albinoism, right? So as we can see in the square here, if both parents are carriers, so they've got one dominant and one recessive, which means that they are normal phenotypes, but they do have the recessive gene to carry on, right? Together as carriers, if they're mating, there's a 25% chance for them to give birth to a child with albinism. Another example of recessively inherited disorder in humans is cystic fibrosis, and this is a lethal disorder if it's left untreated. And the condition is caused by the absence of a membrane protein that transports chloride ions. So the chloride ions result in thickening of the mucous membrane in various different organs that would block nutrient absorption. So for the most part, cystic fibrosis really affects the lungs the most. It makes clearing the mucus from the surface harder, which can lead to a reoccurring bacterial infection, and it would make it very difficult for the patients to be able to breathe normally. The cystic fibrosis recessive allele is actually quite common, especially among the European population. There's about 4% of them being carriers, And again, when two carrier parents get married, they have about a 25% chance to give birth to a baby with the condition and 50% chance to have children that would become carriers of the gene. Okay. So it's estimated that around 30,000 Americans suffer from cystic fibrosis, and 1 in every 31 Americans are carriers for this recessive allele. So even though this could be lethal if untreated, it does still exist in the population. In addition to recessively inherited disorders, there are also genetic disorders that are passed from parents to offspring dominantly. So for this type of disorder, both homozygous dominant and heterozygous individuals would show the affected phenotype. So, for example, achondroplasia, which is a form of human dwarfism, is caused by a dominant allele that would give rise to a mutated growth factor or mutated growth factor receptors, which eventually interfere with bone production. But one thing to keep in mind is the dominant allele that would cause lethal disorder is much rarer than the recessive allele with lethal effect. Since such recessive alleles can be preserved in heterozygous carrier individuals and passed to the next generation, but if you have a lethal dominant allele, it wouldn't be able to survive to reproductive age, and therefore the allele is lost from the human population gene pool. So it's a lot harder for those to sneak through because even as a heterozygote, you would still have that and it could potentially be lethal, versus recessive, you could still be normal if you were a carrier. For certain dominantly inherited disorders, the disease does allow the affected individuals to survive past reproductive age. So that allele would be more common than most other lethal dominant alleles in the human population. So an example of that is an allele that would cause Huntington's disease. Huntington's disease is a lethal neurodegenerative condition whose symptoms only really start emerging later on in life. So when the patient's around 35 to 45 years old. So this is well past the reproductive age, so they could potentially be reproducing and producing offspring that would also have this allele. Although this genetic disorder is still fairly rare. It only affects about 1 in every 10,000 people in the US, but it is something that can continue to be passed on even as a dominant allele, just because it does allow for that person to survive until a reproductive age. So all of these diseases we've discussed so far are kind of the more simple Mendelian disorders, which result from basically one abnormal gene with maybe 1 or 2 affected alleles. There are lots of other human genetic disorders that are what we call polygenic, meaning that they're caused by several genes' additive effects. Okay, so there's several genes that can affect the disease outcome. But thanks to this genetic knowledge, many hospitals today, right, are able to provide genetic counseling to prospective parents who are concerned about family history for specific genetic disorders and the risks for certain genetic disorders as well, especially for these simple Mendelian disorders, because they can be pretty easily assessed using Mendelian genetics. Some of them are a little bit more complicated when you're looking at polygenic disorders, but they're still getting better and better at looking at that as well.